Hello, my name is Jacob Culver from the Mariopan Lab at Yale University, and I am one of the authors of A Second Chance for Protein Targeting and Folding, Ubiquitination and Deubiquitination of Nascent Proteins, published in BioEssays. In this manuscript, we review the intersections of protein folding and protein degradation with a focus on the post-translational modification ubiquitination. We go on to hypothesize a possible ubiquitin-dependent protein folding cycle that may provide nascent proteins additional chances to mature properly. Our interest in how protein folding and protein degradation intersect began during our research of tail-anchored membrane proteins, or TA proteins for short. TA proteins are defined by containing a single transmembrane domain at their C terminus. TA proteins are translated and released directly into the cytosol and must be post-translationally shielded by chaperones and targeted to membranes for insertion. Our now published work in the Journal of Cell Biology shows that a portion of nascent TA proteins in the cytosol are being ubiquitinated. Ubiquitination is a post-translational modification with many functions, the primary of which is to route proteins for proteasomal degradation. Despite the attached ubiquitin chains, nascent TA proteins were not being degraded. So we asked, how can these ubiquitinated proteins with exposed hydrophobicity exhibit stability in the cytosol? And we found that instead of routing for degradation, the nascent ubiquitinated proteins would route through their intended post-translational insertion pathway. With the support of chaperones and presumably cytosolic deubiquitinases, or DUBs for short, ubiquitinated TA proteins are able to be inserted into the ER membrane. Next, the ER-associated DUBs, USP20 and USP33, remove any remaining ubiquitin, resulting in a functional TA protein. This novel finding shows ubiquitination, typically a pro-degradation modification, can occur within a protein synthesis and maturation pathway. So next, we asked, could ubiquitination, chaperone capture, and deubiquitination of nascent proteins occur for a wide range of proteins and not just TA proteins? And could this ubiquitination possibly be a feature of protein synthesis and not necessarily a pro-degradation force? Let's examine this starting at the beginning, when a ribosome translates mRNA into a nascent protein. This linear chain of amino acids must fold into a specific conformation to form a functional protein. The protein folding process is assisted by molecular chaperones. Chaperones are proteins that bind to hydrophobic regions of substrates to shield them from the aqueous cytosol. Ideally, nascent proteins will become properly folded and be routed to various localizations to begin their intended function. However, misfolding still occurs despite the efforts of chaperones. Misfolded proteins often contain exposed hydrophobicity and pose a threat to cells by forming nonspecific interactions or protein aggregates. Chaperones continue to bind misfolded proteins, and the same chaperones that assist in protein folding are often capable of recruiting degradation machinery. Chaperones can recruit E3 ubiquitin ligases to modify substrates with ubiquitin chains. Ubiquitination typically routes proteins for proteasomal degradation. However, we propose that ubiquitination can also serve as a fix-me signal for proteins that need additional chances to fold properly. Next, a portion of the ubiquitinated misfolded proteins will be pulled through the proteasome, which breaks substrates' peptide bonds to recycle the component amino acids. Instead of binding the proteasome, ubiquitinated proteins can also be recruited to unfolding enzymes. Unfolding enzymes such as P97 ATPase can unfold substrates to break intramolecular forces that hold the protein in a misfolded shape and remove some, but not all, of the attached ubiquitin. The unfolded substrate can then be captured by chaperones and interact with deubiquitinases. DUBs are the antagonists to E3 ligases and remove ubiquitin chains to protect substrates from proteasomal degradation. The deubiquitinated and unfolded substrate can then route through our proposed folding cycle additional times. Hopefully, the extra chances will result in a properly folded protein. But terminally misfolded proteins will eventually engage with the proteasome and be degraded. Our proposed cycle of protein refolding relies on ubiquitination and folding status to recruit the machinery required for the next step. Such a cycle would clarify how nascent proteins and misfolded proteins are properly triaged to their intended fates, despite both containing exposed hydrophobic regions and relying on the same chaperones. We look forward to more discussions and research into how pro-folding and pro-degradation pathways intersect to maintain protein homeostasis. Thank you for your time.